I think we are live now. So hello everyone. Very excited to be here again. So today our speaker is Ayo. I've been told that that's enough. For the name. And let me just briefly give an introduction about him. So Ayo was born in Nigeria, but as we just were talking, he actually was also raised in several different countries, which again, I was telling him I'm kind of jealous. But he actually moved in 2003 to US, where he started study, and when he also did the graduate school at the University of Iowa. And then already there at that point, so it got in love with microglia, and in particular with the imaging of microglia. And so that's pretty much what his lab interest is. But before going to starting his lab, he did a PhD in the Dr. Long Kyun Woo lab, first at Reuters and then in Mayo Clinic. No, sorry, that was the postdoc. Sorry, Ayo. So that's actually wrong. So first he was doing his PhD on real-time imaging in the lab of Dr. Michael Daly, and then he moved instead for his postdoc at the Mayo Clinic with Long Kyun Woo. So he actually, again, he studied a lot of different phenomena in microglia. So first focusing on imaging and then looking a lot about the interaction between microglia and neurons and the mechanisms that actually mediate that interaction. And this is part of it is the focus of his lab. So he's currently at the Virginia University and the Department of University of Virginia, at the Center for Brain Immunology and Glia. And there is focus on the role of microglia during development, but also in disorders, for instance, in the cases of seizures and epilepsy. So, Ayo, I mixed up a few things, but I hope that was clear enough about your path. We are really excited to have you here. So the floor is yours. So now you have to share your screen.
Okay. Well, as I share my screen, I just want to tell you, Guy, that it's such an honor and a privilege that that you invited me to be able to give some of our work, talk about some of our work, and you give a very good introduction about my background and my training, and I appreciate that. Again, title of my talk is Microglial Dynamics in Neurodevelopment and Neuropathology. We're at the University of Virginia. We're a relatively new lab, just about 2 years old in August. So what I'm going to be talking about is some of the work that I did as a graduate student, as a postdoc, falling in love with microglia and trying to elucidate some of their interactions and some of the things that they do in the brain. I will most of the work I'll be presenting is published. I'll be interspersing some new data that shows some of the directions that we'll be going into. So this is the outline of the talk. As Guy also mentioned, you know, I identify strongly as an immigrant, being an immigrant not only in one country, but actually in several countries where I've lived. So I think that's why I appreciate the microglia so much, as I'll be laying out in the background. Since I know not everyone studies microglia, I'll give a brief background of them as immigrant cells that take up residence permanently in the brain. I'll go ahead and tell two stories that roughly correlate with some of my work as a grad student and then as a postdoc. And then I'll give some summary and look at some future directions. On that note, this is a general schematic from about a decade old. There's some things here that are not entirely correct, but the general picture here are cells of the hematopoietic lineage on the left and cells of the central nervous system on the right. And although traditionally we thought that these two systems don't interact much, we now know that they actually do interact quite a bit. In terms of the different cell types, you can appreciate here that the neuroectodermal progenitor cells can give rise to all cells of the nervous system except the microglia. And this includes neurons as well as macroglia, which includes astrocytes, oligodendrocyte precursor cells. But the microglia have always been recognized as unique. They're a different cell type. And molecularly, we actually know that they actually show phenotypes that are similar more to these hematopoietic cells that serve as their cousins. Although we also now know that they actually molecular differences between the two of them. So these cells are the cells that I focused on and started studying as a grad student, and I haven't left studying them. Now, in terms of their origins are important. And what we know now from the origin of microglia, especially over the last decade, is that they actually originate early in development from the yolk sac. From the yolk sac, they migrate into the brain directly. This is unlike a lot of other macrophages that also derive from the yolk sac that actually go through an intermediary residence in the fetal liver and then colonize different tissues. The microglia, for the most part, actually arrive directly into the brain. I should point out that there's recent studies that suggest that there may be a subpopulation of microglia that actually migrate through the fetal liver and then take up residence in the brain. Once they get into the brain, they're pretty long-lived. This is data recently that shows chronic long-term imaging of individual microglial cells that shows that actually we can see these individual cells live for a long time without dying in the brain of a mice for up to, in this case, 50 weeks plus. And even at that point, some of them can actually divide and proliferate and persist in the brain. So these are very long-lived cells in the brain. Again, as I mentioned, they come into the brain very early, and we think that this is important. This is a general schematic showing some of the origin of different cell types in the brain. So we have neurons early on being formed in the brain. Microglia, compared to other glial cells, are actually arriving in the brain much earlier at a time point where synaptogenesis, which is critical for the developing brain and the mature brain, is actually ongoing. So there's reason to believe that these cells actually come and as immigrants take up residence in the brain to perform different types of functions. This is a summary of some of the functions that have been identified over the last 100 years, but a very nice review from Amanda Sierra and over in Spain. And as you can appreciate, these cells are actually involved in several features of brain development and brain function. I won't touch on all these features. This is just to highlight the fact that they're known to do all of this. Now, to my interest, 2005 was a significant year for microglial studies, because in that year or prior to that year, we all thought that this is what microglia looked like in the brain, and this is what they did. That is, there were cells that had a central soma and had tentacles or processes that emanate from them, and we're pretty much doing nothing really in the naive brain. Well, in 2005, this study came out first in Science, and another study also in Nature Neuroscience that showed a similar thing, that these cells are actually not static, but actually quite dynamic. And their dynamism is actually a flow off of their process activity. So their central soma remains pretty stationary, but their processes are constantly palpating and filling through the system, the nervous system of the brain. For not only are they doing this in the native state, actually when there's some kind of injury, we can appreciate that the microglia can also respond pretty rapidly in the order of minutes, this is in minutes, to contain that site of injury. And others have actually shown that this is really important to prevent the spread of the toxic substances that occur in the brain during injury. So even these localized injuries, in this case, we induce one, but even when there's a natural occurring injury, the microglia are important to contain that. So I need to emphasize that these studies were done early on, I guess 15 years ago now, and I started grad school in 2006. These studies were especially done in the mature brain, and my question was the dynamics of microglia in the developing brain. So that's a little bit of the background that I'll give, and now I'd like to move into two stories that, again, roughly correlate with some of my graduate and postdoctoral work. I should highlight this first one, that it's work done with microglia, looking at their dynamics in development and in ischemia. I appreciate, as Gaia mentioned earlier, my graduate mentor, Michael Daly, who got me excited about microglia. And so it's appropriate to acknowledge him here. And the excitement that he got me interested in has not left me since. So to study how microglia behave morphologically in the developing brain, it's difficult to look at it in the early developing brain, to look at it with in vivo imaging, because these mice are really small. The brain is really small to perform those surgeries. And also, actually, in the cortical regions that we're interested in to look at in vivo, the population of microglia is actually quite sparse early on in development. So we decided to take an approach of looking at them ex vivo, that is in brain slices. To do this, we take, again, 1 week old, postnatal day 2 to 6 day old mice. excise the hippocampus, cut them up into 400 micron thick slices, and set them in an imaging chamber that's sufficient for them to be maintained for long periods of time. We provide temperature, heat to simulate roughly body temperature at about 35 degrees. And then we can image from anywhere between 3 to 6 hours. And I'll be showing you some of that data in the form of movies. Having done that, so this is a movie here that collates data from postnatal day 2, postnatal day 4, and postnatal day 6 brains, where microglia are highlighted in the white as white cells. These are transgenic mice that express GFP under the control of a relatively unique microglial receptor. And so what I had mentioned and shown to you earlier was that in the adult brain, what is clear is that the microglial soma are pretty stationary, though their processes constantly feel around the tissue. When we looked in the developing brain, what we observed, at least in these slices, was that contrary to what we observe in the mature brain, in the developing brain, these cells are actually quite migratory early on in development. And this migratory ability is present robustly at P2. It actually peaks at P4, and then dramatically between P4 and P6, the degree of migration is actually significantly reduced. I don't show the data here, but if we go ahead to about 9 days old, these cells are stationary just like in the mature system. So we're interested in this finding, and this is just quantification of the data showing that their peak migratory activity is happening at this postnatal day 4 time point. I should say this is especially in the hippocampus that we're looking at. Okay, so this raised the question of what regulates this robust microglial dynamics early in postnatal development that's absent later on in development and also in maturity. And broadly, we can think of potentially 2 factors, though these 2 factors may not be mutually exclusive. We can think of cell extrinsic factors, that is, factors from other cell types in the brain or other extracellular matrix factors in the brain that are released to direct the microglia to crawl and move around. We could also think about intrinsic factors that are present in the developmental program of the microglia. And so my graduate work set out to test at least one of these, which is in the form of apoptosis. Apoptosis is common in development where a lot of cells die, especially neurons, an excess number of cells die. And it is known that the factors that are released during apoptosis can actually regulate microglial activity. So we speculated that apoptosis could be a factor, an extrinsic factor that could be regulating this dynamic early on in development, and so, but not in later development and in maturity when less apoptosis is going on. So we decided to test that hypothesis, and we took this approach to use a genetic approach. But first of all, we wanted to confirm that indeed in our system we see quite a robust apoptosis going on. Again, this is data from transgenic mice that express GFP in microglia. Microglia are shown in green. In red is a dye called PSVU, which labels phosphatidylserine that's present on the outer leaflet of cells undergoing apoptosis. So what we could confirm here was, interestingly, that while you can see quite a bit of PSVU puncta at P2, you can see a dramatic amount at P4, and then it's reduced by P6. And we can quantify that all here, showing that, again, similar to what we saw in our migratory capacity of the microglia, that apoptosis is actually peaking at P4. And these are just examples of these apoptotic structures at P4. Microglia seem to be very interested in these cells. This is a static 3D movie where we are rotating through a section of the tissue. And what you can appreciate is there's a bunch of microglia around. Some of them are actually engulfing this apoptotic material, and others of them are in close proximity. So we speculated that perhaps apoptosis could be regulating this dynamic migratory capacity of the microglia. We decided to test this using a genetic approach, like I mentioned, and we took advantage of the fact that BAX is a pro-apoptotic protein, and in mice where BAX is knocked out, apoptosis is absent. So we wanted to confirm this at first with our PSVU staining. We can show a bunch of PSVU labeling here in the campus of wild-type mice. In the knockout mice, we can see that apoptosis is pretty much abolished in these mice. So again, we can confirm that these guys don't have apoptosis. The number of microglia is not affected in this condition. So we see the same number of microglia. So apoptosis, at least at this stage, does not affect microglia at P4. And the relevant question is, what about their migratory ability? I was somewhat disappointed to find out that indeed we do not find any regulation of the dynamic activity of the microglia by Bax-dependent apoptosis. So in littermates that are wild type or Bax negative, we see the same degree of migration going on. We see the cells moving at the same velocity. They travel roughly the same distance, and even their motile process activity seems to be unchanged. So our data suggests that at least Bax-dependent apoptosis does not regulate this microglial phenomenon. And that's where I stopped in grad school with this project. But I still have to remind you that although we've seen at least one aspect of apoptosis, we've not tested other factors that are potentially cell extrinsic. For example, the extracellular matrix, factors in the extracellular matrix, or even other factors that are released in other processes during development. I'd be happy to hear suggestions on this, by the way. Now, however, something that has been interesting is to ask the question whether intrinsic developmental programming factors may be regulating this microglial dynamics. And at this time, a postdoc joined my lab a year ago, Kanchan, and she's starting to work on this project. And what Kanchan did was took advantage of what we know in the literature to do some long-term imaging of microglia in vivo. And the premise that she had with this, the premise in a second, but what is known in the field is that you can apply this this drug, and it will efficiently eliminate the microglia, shown here in green as green cells. Gradually over time, with the application of this drug, you lose the microglial density, so you can kill them off. And once you take the mice off this drug, you can actually start seeing the microglia repopulate or come back. What was known in the field from actually last year, studies from Ligan's lab, was when we look at the transcriptome of microglia in early postnatal development at the time point that we had just described at P4, and you look at it 4 days after this repopulation, during this repopulation period in the adult brain, they actually share an overlap of a significant number of genes. And so this intrigued us that maybe we could ask the question of intrinsic developmental programming factors by performing an experiment where we deplete the microglia in the adult brain, so the brain is in the mature state, having the same extrinsic factors in the mature state, but when we deplete them and let the microglia to repopulate, we could drive them into showing this migratory ability because they've gone back to the developmental programming state. At least the microglia have gone back to that state. So we decided to test that hypothesis. And to do this, we do these chronic in vivo imaging studies where we can make a craniotomy in a mouse, implant a window in the mouse, and we can monitor the microglia immediately. And sometimes we do those studies. But the problem is because microglia are sentinel cells, and as I showed you earlier, they respond to even the smallest amount of injury. And this craniotomy procedure is actually injury-inducing. We actually use another approach to implant the window and let the mouse recover from that injury over a period of 2 weeks. The microglia get initially activated, but then they get deactivated to somewhat normalcy. And then we follow these cells in this condition. When we do this, what I'm showing you here are some of these microglia in the control brain. And you would remember that as I showed you earlier, these cells are actually pretty dynamic in terms of their processes. But if you follow on track their cell bodies for extended periods of time, they're actually pretty stationary. And this is imaging done over the course of 5 to 6 hours. They're actually pretty stationary. That's not new. We know all of that. We now ask the question, what about when we repopulate the microglia? Do they take on this phenotype of increased migratory ability? And indeed, what we find out is that they do. So I can show you this big, large scale movie to show you some of the cells. For example, you can appreciate some of these cells crawling around and moving around. You can see these here. And we can actually zoom in and focus on some of these cells with these movies here. For example, this cell moving and crawling through, this cell crawling down. So we see an increased dynamism of these cells in terms of the cell body translocation in the brain, even though the brain is adult brain. So we think that the intrinsic factors in the microglia allow them to actually distribute themselves and move. And while we haven't done extensive studies, and Kanchan is in the beginning stages of this, begin to address this question. We see that later on, if we wait for about a 2-week period, we don't see as much migration in the microglia. So they seem to go through the same developmental program. And here I show another example, a little bit slowed down, to show some of these cells that are dividing. Once they divide, they actually start to migrate away from each other. So this is, again, another feature that's typical of the developing brain, but not of the adult brain, unless we do this depletion repopulation studies. So we're still pursuing this, trying to quantify some of this on a time course, on a time scale to begin to answer this question. But we think that these are promising results to at least suggest the involvement of intrinsic factors. The next step would be potentially to look at what are the specific intrinsic factors or molecular factors that are driving this activity. And we're excited to pursue some of those studies. Coming back to development, what we also wanted to ask was not only what are the microglia doing in their normal state in development, but even in pathology as well. So we, in my graduate work, I was interested in ischemia, which is pretty common in the developing brain. So what we did was we asked this question of, a lot of work had been done in microglia, in ischemic insult in the adult brain, but we're interested in the ischemic insult in the developing brain. And I found these results are pretty fascinating and something that we still want to pursue. What happens is that in the developing brain, if you look at a P2 slice, and you would recall that in the P2 stage, the microglia are actually quite migratory. So if you look at cells in this state and put them in a simulated ischemic condition where we deprive the tissue of both oxygen and glucose to simulate ischemia, and we do that also in a P6 brain slice, it's remarkable what we observe. So if we focus on the P2 stage, you can see that these cells, despite being in an ischemic context of no oxygen and no glucose and presumably very little energy, these cells are still dramatically mobile, and they are crawling around the tissue. I followed the tracks of only 2 of these cells over the course of 6 hours. On the flip side, when we look at P6, just a few days later, these cells are not moving. Actually, we start to observe something that we hadn't intended to see, but we noticed, which was that some of these cells that express GFP, like some of these cells here, they actually start to blink off, which we suspected was an indication of these cells dying in this context. We never see this happen at an earlier stage at P2, which makes us actually intrigued that there may be a developmental difference between the microglia, these very short time points that allow the early postnatal microglia to be less susceptible to ischemic insult compared to the later postnatal microglia. And we still don't know the answers to those questions, but we're interested in pursuing them. What we did come up with, though, was the question of microglial cell death. And to our knowledge, this had not been investigated in the past. And we thought that we had come up with a nice assay to be able to visualize this and quantify this in real time. So again, this is in an OGD condition, which is oxygen glucose deprivation. This is a transgenic mouse that expresses GFP in microglia. And in this tissue, the tissue is bathed in a context of cytox, a dye that labels and binds to DNA. But this dye cannot bind to DNA unless the DNA membrane is compromised. So what you see as I play this movie is that this is 2 hours of ischemic insult, which, by the way, at this time point, a lot of neurons are already dead. The microglia are actually quite resistant, but they eventually die as well. And what you see here is gradually the cell soma would swell up and then eventually lose their GFP while they retain and get the cytox, indicating that the membrane is compromised to release GFP as well as take in cytox. And so that's what this movie shows. So you can see the swelling of the cell gradually, and then the loss of GFP and the uptake of cytox. So we think that this is a good assay to measure cell death or look at cell death. And this has become very interesting because in terms of the microglial role in ischemia, both in development as well as in adulthood, it seems like microglia are playing a neuroprotective role. At least that's what a lot of the data coming out is showing. So there are several studies here showing that when you eliminate the microglia, you actually get a worsening of the seizure outcome. And conversely, when you actually introduce microglia, you actually get an improvement of stroke outcome, even when you do a trans-species from human microglia to rat. So this is exciting to show that perhaps microglia could be used or harnessed as a therapy in ischemic injury. Now, in order to be able to do that, presumably we have to preserve microglial survival. And to understand how to preserve microglial survival, we have to understand the mechanisms that that microglia undergo to lead to their cell death in this ischemic context. And that's what my next focus was in this project, was to ask the question, what regulates microglial cell death during ischemia? Well, we took advantage of the well-known idea that there's a calcium overload hypothesis, where a lot of cells die during ischemia because they have an excessive influx of calcium that leads to downstream cytotoxic mechanisms. So this is a movie compilation of 3 different conditions. First, a control condition that has normal calcium present, then an OGD or ischemic condition that also has normal calcium present, but then a later condition that is OGD, so ischemic, but lacks extracellular calcium. And what you'll observe if I walk you through each of these is that you can appreciate that the microglia in this state of roughly about P6, P7 are actually quite dynamic. They're happy, they're moving around, they're crawling around, they're moving their processes. But if we look in the ischemic state, they lose that activity physically, but they also start to blink off like we've recently shown and discussed. And so they start to show evidences of swelling up and blebbing and actually dying over the course of 6 hours. We get roughly about 40% of the microglia die during this 6-hour period. However, when we take away the extracellular calcium, even though we have the same ischemic condition, we preserve pretty much all of the microglia do not die in this context, even though they are significantly hampered compared to the control condition in terms of their motile activity. Okay, so we come to the conclusion that calcium is really required, or calcium influx is really required for this ischemic death in the microglia. Next, we have to ask what precisely the mechanism of calcium influx. And it's well known that purinergic receptors, especially ionotropic purinergic receptors, can regulate calcium influx. And we know that ATP, which is a purinergic signal, is actually released at high amounts in the ischemic context. So what I did was test the hypothesis that the P2X7 receptor, which is a unique purinergic ionotropic channel that actually is gated by ATP and functions to allow ions to come in when it's activated at low ATP concentrations, but at high ATP concentrations, it not only forms an ion channel, but actually forms a very dilated pore that allows a massive influx of calcium, leading to swelling of cells and eventually cell death. We wanted to study this in a tissue context for the microglia to ask the question whether P2X7 receptors could be regulating this feature. We've done these experiments using pharmacological approaches, and we get the same result as when we do a genetic approach. But I show you the data here using a genetic approach where we have heterozygous mice that have 1 copy of the P2X7 receptor and knockout mice that have no copies of the P2X7 receptors. And these are littermates that we subjugate to the same ischemic condition at the same time. And what you can appreciate here with these yellow highlights are cells that blink off during the context of ischemia, and so they die, and there are a massive number of these in the head condition. And although some die in the knockout condition, it's a significantly reduced number of them that die. Again, we don't think that the P2X7 receptor is the sole receptor responsible for this ischemic cell death. We think that we show that only about 50%, with extracellular calcium reduction, we get rid of the cell death by 100%, pretty much. But with P2X7 antagonism or genetic abrogation, we only get rid of it about 50%. So we think that there are other factors that could be regulating this calcium influx, and we're interested in pursuing what these factors are in the ischemic context for microglia. What is interesting is that others have also used this approach to target P2X7 receptors, especially pharmacologically, and show that this actually improves recovery from ischemia. The problem with some of these studies is that P2X7 receptors, according to the data, is actually kind of promiscuously expressed by different cells. In the mouse, the RNA-seq data suggests that microglia are principal expressors of these, but also oligodendrocyte precursor cells also express this. And in the humans, although microglia express it highly, oligodendrocytes also express it. So what we are thinking about is future studies to look at potential ways in which we can specifically target microglial P2X7 receptors in an ischemic context and see if that would actually improve the phenotype, because these results do not clearly distinguish whether it's a microglial specific role or other cell type specific role. Now with that, I'll just summarize this part of the talk, which again corresponds roughly to some of my graduate work in this, showing that microglia exhibit robust microglial capacities in early development that is rapidly downregulated. We began to investigate some of these mechanisms. We show that apoptotic mechanisms at least don't seem to be a primary driver for this. And we think that there might be some intrinsic features and factors that may be regulating the developmental program of microglia to drive their migratory abilities early in development. Secondly, in terms of this ischemic injury, it's something that we think is fascinating because we see that there's a very early on at P2, which corresponds to the in utero period in humans, there's a really high resistance of these microglial susceptibility to ischemia, and we still don't understand why that is. Presumably, it could be because of some metabolic difference with microglia at that time point, and we're interested in investigating that further to use as a way to potentially also protect microglia from cell death. Apparently, it could also be a developmental program that's changed with this, but it'd be interesting to look at potential metabolic differences between microglia at these unique developmental time points. We also have this assay that we think can be used to assess cell death in vivo, or at least in tissue, in slices, and especially in vivo, not only for microglia, even though that's our focus, but also for other cell types. Finally, we have identified at least the P2X7 receptor as a potential target to reduce microglial cell death, which we think if we can preserve microglial survival, then we can actually improve in ischemia. And we're looking at future studies to do some of that. I'll switch gears now to move on from my graduate work to some of my postdoctoral work, investigating microglial-neuronal interactions in hyperactive conditions, especially in seizure pathology. Again, to appreciate, this is work that was done with my postdoctoral mentor when we're first at Rutgers University and then moved on to Mayo Clinic, and he's still there at Mayo Clinic now. So at the time, as I was starting my postdoc, it was clear that microglia, there were novel findings suggesting that microglial dynamic activity is not only directed randomly through the tissue, but it seems to be directed also specifically to neuronal elements. And so these are studies from as early as 2009 to about 2012 when I was finishing grad school and about to start my postdoc, that actually started to indicate that microglia are physically interacting with neurons. So the phenomena was present, and we've summarized some of this. A graduate student in the lab has summarized some of this if anyone's interested in a recent review from last year over the last decade of these interactions. And what we're interested in was potentially the mechanisms that are responsible for these interactions. So I started my postdoc with the idea of driving neuronal activity to see if we can see morphological responses in the microglia and use that as a paradigm to try to decipher and determine the mechanisms underlying it. So this is work done especially in slices that I'm showing here, both in the hippocampus and in the cortex, although we focus most of our work in the hippocampus, and I can explain why that is. We use glutamate as a neuronal hyperactivity driver to increase the level of activity going on in neurons and communication between neurons to see if microglia listen in. I should say, although I don't show the data, microglia are electrically silent to glutamate, at least at the inotropic level. They do not respond electrically to glutamate treatment. So any responses that we see should be presumably through other cells that respond directly to glutamate that then signal to the microglia by some other mechanism. And that's what we're interested in looking at. So here's the hippocampus. This is the pyramidal layer of the hippocampus where there are a bunch of neurons present. They're not labeled here, only the microglia are labeled. But what we do is we bath apply glutamate into the system. And what you can appreciate here is that while there are few microglial cell bodies and processes, what you can appreciate is within 15 minutes of treatment, you can appreciate there's an intense number of the microglial processes present in this region. We can color code this in a green and red setting, where red indicates the initial time points and green indicates the later time point. And you can appreciate that there's a lot of green in this context and in these regions where neurons are populating. So we think that the microglia are responding to this, and I'll show you a movie of that in a minute. This is not only true in the hippocampus, it's also true in the cortex, where you can appreciate that there's a lot of green processes that are newly developed and spread out, radiating from the cell bodies of the microglia. The reason why we choose the hippocampus is because the hippocampus has this nicely layered structure where we can see the processes and be able to measure it and quantify it for future studies. We not only see this in slices, we actually confirm that we see it in vivo as well. At a somewhat longer time scale, in this case, it takes about 40 minutes, but you can appreciate that after 40 minutes, you can see that some of these processes are actually extending from the microglia in green. And we can quantify this here where we apply, this is in slices here. When we apply glutamate at this time point, we can start to see a response of the microglial processes as they increase what we refer to as the extension index. They increase the extensions in response to that phenotype. Now to show, let's see, I hope this works. Okay, to show some of this in a movie format, again, this is what it looks like. These neurons are present in these regions, which I'm not showing you because they're not labeled. But you can appreciate that microglial processes are not robustly present here. And as I play the movie, when you see glutamate is added, what you can appreciate is the processes will all converge in this region. And so that's it. Glutamate's added, and you can see a lot of the processes start to converge in there. Glutamate is added, a lot of the processes start to converge in there. What we wanted to confirm was actually that we can see that the microglial processes are converging in here, but we asked the question, are they really targeting neurons, or are they just randomly converging in there? So we use a transgenic approach, using double transgenic mice that have microglia labeled in green, and neurons in this case labeled with YFP, but I'll be showing you it in red. So we can visualize both microglia and neurons at the same time. And we're doing this again in the hippocampal region here, which I've blown up to show that within 13 minutes in this case of glutamate treatment, you can appreciate that there are these microglial processes that actually start to make contact with the neuronal elements. We can look through a Z-stack to confirm that these are actually directed specifically to the neuronal cell bodies. We also see this targeting the dendrites of the neurons. We focus on the soma just because the soma is the largest surface area for us to quantify. And when we quantify it, we see that there's an increase in the presence of glutamate with this microglial contacts of the neurons. So we think that we have a paradigm now that we can use to interrogate the mechanisms by which microglia use, at least glutamate-dependent mechanisms by which microglia use to interact with neurons. So we decided to use a pharmacological approach in brain slices, well-established pharmacological approaches, applying glutamate, and then using blockers, known blockers of different glutamate ionotropic receptors. In this case, you can see again, to remind you that when glutamate is added, we can see a lot of the green present in this region, and we can quantify that here with the dark green bar or dots, that once glutamate is added, we see an increase in the microglial process extension to that region. When we add CNQX, which is supposed to block AMPA and kainate receptors, we can still see an increase in the green present in this region. And we can quantify that there. That it seems like blocking AMPA and kainate receptors in the presence of glutamate does not affect this microglial activity. However, when we apply AP5, which is known to block specifically NMDA receptors, ionotropic NMDA receptors on neurons, we see that that's abolished here, where we don't see a lot of the green, and we can quantify that here, where the result is, even in the presence of glutamate, we get a flat line. So this is somewhat of an antagonist approach. We can use an agonist approach where we apply these drugs, including NMDA itself, and we can induce the phenotype. And this can be shown here in this red, an increase in the extension of the microglial processes. When we apply kinic acid, which is supposed to directly activate AMPA and kainate receptors, we don't see an increase of those, shown here in the dark red. And then when we apply NMDA to certify that this NMDA is acting directly on NMDA receptors, when we block NMDA receptors with AP5, we again get another flat line and not a response of the microglial processes. So we've identified that NMDA receptors, presumably directly on neurons, since microglia themselves don't have functional NMDA receptors, is driving this phenotype. NMDA receptors are well known to be extremely calcium permeable, so we wanted to ask the question of whether this is calcium dependent. So we performed the experiments again in a calcium deficient media, And we see that we can essentially block the response, leading to no green processes present in this region. So we see that this phenotype and this communication between microglia and neurons is actually calcium dependent. Finally, all of this is looking at it from the neuronal side, because it's the NMDA receptors and the calcium influx is being derived from the neurons. What is regulating it? What is regulating the microglial response? We were also interested in. And at the time, we knew that there was a certain receptor called the P2Y12 receptor, which is uniquely expressed by microglia, as shown here, where this immunohistochemistry for P2Y12 is present on the microglia, all in green. And so we asked the question, when we use mice that are deficient of that receptor, do we still see a response to glutamate treatment, or NMDA treatment in this case? And when we apply NMDA, you can appreciate that there's no green present in these cells, and so they do not respond. While wild-type mice respond, to NMDA, P2Y12 knockout mice in the kind of magenta actually have a flat line. And so we arrive at the conclusion of this mechanism that when communication between microglia and neurons actually happen when neurons release glutamate, for example, as an excitatory neurotransmitter, that leads to activation of NMDA receptors present, specifically NMDA receptors present, leading to calcium influx. And although we've not yet identified the full downstream mechanisms, somehow this leads to the release or efflux of purinergic signals like ATP. We still actually don't know the avenue of release. It could be through some other channel. It could be through some vesicular release. We've not identified this in this system, and these are still outstanding questions. But once ATP is released, it binds to P2Y12 receptors on the microglia, and then the microglia respond and target the neurons. And we think that these are interesting results. Now, these are phenomena that we describe as a basic science level, but we wanted to know if this is relevant for pathology, for various pathologies. And so we focus, for example, on seizure disorders, because seizures are actually quite common. They're ubiquitous. They affect all demographics of age, sex, race, ethnicity. And previous work in the seizure disorder fields and epilepsy in specific is actually focused on neurons. Well, now we know and appreciate that these are the cells that are present in the brain that are actually regulating and functioning to provide sufficient proper functioning of the brain. And the neurocentric mechanisms that have been developed in these seizure contexts have been insufficient clinically to treat the condition of repeated epilepsies or repeated seizures or chronic epilepsies in about a third of patients. So there's still a lot of work that needs to be done for us to presumably tackle the seizure disorders. And largely this has been ignored. Other cell types have been ignored. And since we are especially excited in microglia, we thought that we could study this and study these interactions between microglia and neurons and the functional significance of them in this condition or in this context. We needed to use a seizure model, and so we used the chemoconvulsive seizure model, where you can inject either intraperitoneally quinic acid, that is a glutamate analog, and it induces seizures which can be scored behaviorally from a scale of 0 to 6, or actually 7, where the mice die. When you inject it intraperitoneally, there's a gradual increase in the seizure activity of the mice. When you inject it directly into the brain intracerebroventricularly, you can get a pretty rapid response. Nevertheless, in both of these responses, we were asking the question, what is the microglial phenotype? What is the microglial phenotype earlier on in this seizure context? So we sacrificed mice at about this 45-minute period where we see robust seizure activity in the mice. And while in control mice, microglia have very few primary processes emanating from them, you can see that both with ICV and with IP, they show a dramatic increase in the number of processes, reminiscent of what we saw in the context of glutamate treatment, where they produce a lot of processes directed towards neurons. So we think that this is a system that recapitulates, a seizure system that recapitulates what we saw in our basic studies of glutamate. We can quantify this number to show that they're significantly increased. And I don't show you the data here, but we've also confirmed that when we block NMDA receptors in this context, we can also reduce the microglial phenotype in here. So we know that this time a postdoc joined our lab as well, also last year, Kaushik, beginning to address, trying to address some of these questions of whether this can be relevant for seizures and microglial roles can be relevant for seizures. And he took an approach that I mentioned earlier where we can apply some of these drugs that actually eliminate the microglia. So here it eliminates the microglia, leaving the brain mostly intact for what, at least structurally intact from what we can see. The number of neurons are not affected. The number of astrocytes are not affected. The vasculature broadly is structurally largely similar, but the density of the microglia, actually is reduced significantly after a week. And in that context, we can now induce a seizure. And interestingly, when we do this, what you can appreciate is that in control mice, in control littermates that were exposed to a controlled diet where the microglia was not eliminated versus the littermates that were exposed to a diet where the microglia was eliminated, after the first 2 hours, you don't see much of a difference between them in their seizure activity, but subsequently at the second and third hours, at the beginning of the third and 4 hours, you start to see a significant difference in the seizure activity. And it seems like the generation of seizure activity is not affected in microglial deficient mice or mice without microglia. But it is actually the calming down effect that results afterwards that seems to be perturbed in these mice that lack microglia, suggesting that microglia are providing some kind of calming effect to the neurons. And we're interested in pursuing this further to understand the nature of those calming effects. Given our basic studies where we saw that P2Y12 receptors are responsible for this increase in the microglial process dynamics in response to high glutamate, we actually asked the same question in P2Y12 knockout mice, whether the degree of seizures is affected. And we see remarkably that both in the IP injection condition, as well as in the ICV injection condition, the severity of the seizures are much worse when you lack P2Y12, which is, again, I should remind you, specifically on the microglia in the brain. So in both these conditions, you get worse seizures. The onset of seizures is actually dramatically earlier without this receptor on the microglia. And the mortality rate, that is how much the microglia die, or how much the mice die, is actually significantly increased in these P2Y12 deficient mice. Morphologically as well, we see an increase, like I mentioned earlier, in wild-type mice that is significantly reduced in knockout mice in a seizure context. So we think the significance of this is that microglia, at least through its P2Y12 receptor, may be providing some calming effect on the neurons, and we're interested in pursuing this further to try to identify the specific mechanism by which they do this. I'd like to use the last maybe 5 minutes to end on telling you a little bit about another side of the lab that we're focusing on this, to look at microglial interactions in vivo. So these are studies where we were interested in looking at the dynamic activities of microglia and the interactions with neurons in vivo. Again, you've seen some of these images of microglia being pretty ramified in the normal state. However, when we look about 6 hours of seizures, we start seeing them show some of these interesting phenotypes of what I'm referring to right now as pouches. So they show these structures present. We can see it at 6 hours, we can see it at 24 hours. We now ask the question, what are the targets of these? And we speculate that perhaps they are targeting neurons in this, and maybe potentially even phagocytoses in the neurons. So we use our double transgenic mice. Again, you can appreciate some of these structures present at 6 hours and then at 24 hours as well. When we overlay this with neurons labeled, at least the dendrites of neurons, you can appreciate these beaded swellings that we're not the first to describe, but others have described this in different pathological contexts. And so you can appreciate them. What we show here, though, is that the microglial pouches that we identify here, for example, are specifically geared towards these swellings. So we thought this was interesting, another phenotype of microglial neuronal interactions. And we asked the question, what is the dynamic of this activity and interaction with the neuron? So we followed these structures over a long period of time, in this case 4 hours, We've done this for up to 6 hours. And what we show is that, remarkably, contrary to what we would have expected with the well-known feature of microglia being phagocytic, we're actually seeing pretty little phagocytosis in this case, or at least engulfment of these materials. So this is an extant pouch that's present at the beginning of imaging, which is roughly about 6 hours from the seizures. And we can see that they are maintained for an extended period of time without resolution. We can see some of them are actually newly formed in yellow, which are not present initially, and then they are newly formed, but then they also maintain for extensive periods of time. You may appreciate that some of these shrink. So this starts out large, but then it shrinks to this standpoint, and I'll come to that in a second. But let me highlight the few of them that we noticed that are actually engulfed. And this occurs rapidly, for example, between the 2-hour and 3-hour period of imaging here, where we show that this is rapidly taken up by the microglial cell soma. And I should point out that these are actually a minority of the processes. So most of the processes are maintained over this long period of time. A few of them start to move towards the cell body, and a few of them are new. But here I would like to show you quickly a time-lapse movie of some of these processes, just showing you several of them that over the course of, in this case, 4 hours, you can appreciate some of them shrink. As you can see here, this is one of those that's actually engulfed rapidly, but this is a minority of the processes. Here you can see that most of them are maintained extensively. I'll let that movie play again quickly. It's sped up really fast, but you can appreciate that a lot of these pouches are intact. So given that we don't see phagocytosis or major phagocytosis here, and we can also confirm by our dual channel imaging that as the microglial processes shrink, in this case, you can see over the course of an hour it shrinks. We also see the shrinking of this beaded dendrite in the neuron over time. Okay, so we asked this question using our chronic imaging approach. Can we look at these structures at 6 hours and then follow the fate of them, of what happens to them 24 hours later? And that's exactly what we did. And forgive me that this is a little busy, but I'll try to walk you through this. So we can see some of these beaded swellings that are not attracting microglia. And for whatever reason, majority of them don't attract microglia. So we can see some of them here. And we can ask the question, what happens to these guys that microglia are not attracted to for whatever reason? And what happens to them 24 hours later? And we can see that they are maintained. They're not resolved. But these guys that had microglia attracted to them actually eventually become resolved. And so the dendrite that was once beaded is not beaded anymore in there. And we can quantify this. While some dendrites that lack microglia, while some swollen dendrites that lack microglia can be resolved, only a minority of them can be resolved. Conversely, the microglia that the beaded dendrites that actually have a lot of microglia, actually a high significant number of them get resolved. And we're working on testing this hypothesis more, but what we are seeing, or what we're proposing, is that these microglia are actually functioning to heal these beaded dendrites. Understanding some of the mechanisms could be instrumental in understanding how microglia can be used to target the resolution of structural deformities in ischemia and in seizures, and potentially other pathologies where we see these phenotypes. So that's a summary of the work we've done. I'll just like to summarize right here and to remind you some of the things that I highlighted. We talked about development and microglial dynamics of this rapid migratory ability in development, or rapid downregulation of these, and we're working on some of the mechanisms there. We talked about some of the interesting phenotypes of microglia in ischemia, and especially their death, regulated by P2Y12 receptors, and working on figuring how to specifically target microglia in this context and the consequence of that in an ischemic behavioral context. In the second part, I talked about the signaling mechanism and understanding some of the signaling mechanisms that goes along with NMDA receptor signaling from neurons, targeting P2Y12 signaling in microglia, and the relevance of that in neuroprotection in seizures. We've also recently identified this novel structural phenotype of microglial pouches, and we're working on the mechanism by which microglia may be resolving some of this. So that's the summary of the work that we're interested in. Our lab is, we think of ourselves as the microglial lab with a microglial focus. We think pathology is important, but we work on pathology from a mindset of looking at development, because we believe in the dictum that to better understand how things fall apart in neuropathology it's important to appreciate how they are put together in the first place in neurodevelopment. So that's why we harness both development and pathology. And so far, what I've shown you is work that we've been interested in ischemia and epilepsy or seizure disorders, but we're also interested in other sorts of pathologies, looking at the vasculature and vascular pathology and physiology, as well as neurodevelopmental disorders. I haven't shown you any of that work, but there are folks in the lab that are working on this. Again, we're a relatively new lab, but we're a very excited lab. We refer to ourselves as the Microglial lab. I've shown you some of the work that's being done by 2 postdocs in the lab. There are, of course, other members of the lab that are doing other things, both grad students, technicians, undergraduates, that they're worthy of me to appreciate here. And someday maybe I'll get to talk about some of their work. And I also would like to appreciate my family that makes studying development not only fun in the lab, but also at home. They're giving me the time to be able to be here and to present some of this. And also our center that is a magnificent center and provides a lot of engagement and stimulation, our department, a lot of support for a new lab like myself, and especially our funding sources, both external to UVA as well as internal to UVA. And I'll be happy to take any questions at this point.
Thank you, Ayo. This was fantastic and very exciting, and you really passed that excitement, I think. And there are indeed many questions, so I think I'm gonna go through them.
Yeah.
First of all, thank you for this very interesting talk. So do you know if microglial cells that repopulate the brain after depletion have the same transcriptomic profile of the resident one before depletion?
That's a great question. So this is work that was done in, at least Lee Gan has done this work. You can see it in a PLOS biology paper, where she looks at early in repopulation. Again, like I mentioned, they are similar to that in development, but then they mature eventually to look like roughly the same as the regular microglia maturing. But that takes at least a month for that to happen. Good question.
Great. Next one is, again, thank you for your talk. So in our work, we see microglia engulfing neurons without those being in apoptosis. So we believe that in some brain regions and ages, microglia may receive a neuronal signal to perform phagocytosis that is not related to cell death. Do you have any idea on mechanisms that may mediate this interaction?
Wow, that's really interesting. So I'm a little confused about I guess I assume that if there's phagocytosis, it implies that there's cell death, because when you eat something up, that thing is not around anymore. Maybe I'm wrong in that assumption, but what I would say is what I am coming to, especially with the last part of my talk, where I showed some phagocytic-like structures around neurons or neuronal elements that doesn't lead to the death of those elements. I would agree with this questioner that I don't think everything that we see as phagocytic actually ends up being a clearance effect or a clearance phenomena. What we're seeing in our data is that you can see structures that look phagocytic, but they may not end up being engulfed, if that makes sense. And I would agree with that comment.
So next question is, how do microglia sense the presence of other microglia such that tiled non-overlapping domains form?
Yeah, there's really been, we know that they do this because they avoid each other. I will say this, though, that actually what we see in the repopulating phase is that they do that less. That is, they do actually seem to physically climb on each other a little bit more during the developmental repopulation and during the developmental stages. So, but we don't know what the factors are. To my knowledge, nobody has identified some of these factors, but we know that they are there. And this is why we're going to have a job for a long time trying to figure out some of these questions.
Yes, another question from the same person actually is, do microglia cross anatomical boundaries during development, so similarly to what happened to migratory interneurons, or are they confined to certain regions, so similar to the cortical pyramidal neurons?
So I don't know what is thought of as regions, right? I don't know what constitutes a region. Are we talking about cortex versus hippocampus? Can something in the hippocampus crawl into the cortex? Or can something in,
it could be also
the
very
auditory?
so because it refers to interneurons and then pyramidal neurons. So I don't know if it's also.
Yeah, so there was some, I need to, I've not focused on this work since grad school, but I used to read some studies that talked about tangential migration of the microglia early in development. I don't know the degree to which that is, how that is done, but especially in the developing brain, I would expect that some of that is going on because they have to populate the whole brain, right? And so there's potentially a gradient by which they populate the whole brain. We know that there is a gradient when you deplete all of them and repopulate. We can see a gradient of this. And that may be a gradient that's recapitulating what happens in development. So something to that extent. But that's an interesting question as well.
Next one is about whether extrinsic factors in different brain regions.
Yes.
So how those regulated microglia behavior, and a follow-up question is whether do you know if you implant microglia from one region or graft microglia from one region to another, how they behave and whether they behave differently.
That's a fascinating. Those are fascinating questions. So the first one, if I remember well, was extrinsic factors in different regions. Actually, again, we don't know what these extrinsic factors are that will be regulating this. I have not identified any of them yet. What we do know is that from my work, which is focused on the hippocampus, I see this. I described to you guys the early postnatal peak and then reduction of the migratory capacity of the microglia. What others have shown is looking at it in the cortex. And in the cortex, that peak and reduction actually is present earlier on in embryonic development. So about E14 to like E18, you see that peak and reduction. So we see a difference original difference in these capacities of the migration of the microglia. So presumably, if there are extrinsic factors? I'm not convinced that they are extrinsic factors, but if there are extrinsic factors, we could imagine that the extrinsic factors are regionally regulated. And then the second question, I believe, was about microglia engrafted from one region to another region, what would happen? I'm not aware that those studies have been exactly done. What I can say is groups like and Schaefer's group have actually shown that microglia in different regions, and Lindsay DeBise also shows this, that microglia in different regions actually have regulated by local cues. So for example, in the cerebellum, microglia behave slightly differently than in the say cortex or hippocampus. And even in the substantia nigra, different regions of the brain, they have different phenotypes and actually molecular expression. Now, has anybody taken microglia isolated from the cortex and put it in the cerebellum to see if the cerebellum, if they'll take on the phenotype of the cerebellum? That's an interesting question. I'm not aware that that's been clearly done. At least I can't think of it off the top of my head. But those are great questions.
So there is another question. I mean, it's all along the same line, basically. So they were asking whether hippocampal microglia is different than from cortical microglia, but then there was a specific part on whether they have different response to calcium. So I don't know if you want to add something on that.
I'm not sure what exactly they mean to calcium. But
I don't know. I mean, I don't know if it calcium fluctuations. That's what I think.
So what I would say is that I've done studies, imaging, at least in slices, both cortical microglia and hippocampal microglia, and basic responses to, for example, glutamate, to, for example, taking away extracellular calcium, are roughly the same. Basic responses are roughly the same. What I'll say, and I've never really quantified this, is that once you cut a brain slice, you'll actually observe the microglia actually start getting activated. So they start shortening their processes and they start getting bigger in terms of their cell bodies. What I have noted, and I haven't quantified this, so this is kind of not, this is a basal impression, is that I see that it seems to me like cortical microglia activate much faster than hippocampal microglia. So if you look at the difference in the morphology of cortical microglia, say 2 hours after you've cut the brain slice, they are very activated. But if you look at it in hippocampal microglia, you might have to wait 4 or 5 hours before you see them at the same activation, morphological activation state as the cortical microglia. So I think that there are differences, at least in these responses. I don't know about calcium responses specifically.
Yeah, also staying on the calcium part. So there is a question about your OGD model.
Yeah.
And whether you have any clue about the cellular source of the extracellular calcium, the signal of the MG cell death.
The cellular source of the extracellular calcium.
Exactly.
I do not know. I think there's a bunch of extra. I mean, I'm trying to remember. Well, it'll be hard to determine some of that because, you know, the brain is bathed in a sea of extracellular calcium too, you know, constantly being released. And I don't know if any, I wouldn't be surprised if it's not from one cellular source. I would imagine all different types of cells are the source of the extracellular calcium.
So maybe one last question. So do you know what are the clinical implications of inhibition of postnatal microglia apoptosis?
The clinical implications of inhibition of postnatal hippocampal what?
Microglia apoptosis. Not hippocampal, but microglia.
Okay. Yeah, okay. That's a fascinating, really interesting question. We do not know. And a lot of the studies that have done away with microglia, so they've eliminated the microglia, have been done especially in the adult context. And one of the things we know about microglia is they're especially important in the developmental context. So microglia have done what they needed to do during development, and then we deplete them in adulthood, right? When we inhibit, so we know that the microglia undergo apoptosis roughly about the 3rd week of postnatal development. I don't know that anybody has inhibited that apoptosis at that time point to see what the effect is. And that
what leads to prolong that window, I guess that's in part the question, maybe.
And by prolong that window, you mean keep to delayed. Yeah. I don't know that anybody has done that. Now what we are interested in is kind of a similar thing to that question is with these Bax knockouts. The interesting thing was when I did the Bax knockouts, I focused on it only in the early postnatal period, right? And microglia are not undergoing apoptosis then, but later on they are. We can now ask the question, what's the impact on this Bax-dependent apoptosis on microglia? Does it actually change their number? And then if it does change their number, can we selectively do that to the microglia, preserve their numbers, don't reduce their numbers, and ask what's the consequence of that? I'm not aware that it's a good question that the questioner asked. I'm not aware that there's any studies that address that. I think that those are extremely fascinating studies because microglia do undergo cell death, but we don't know why. We don't know why they have to undergo the cell death. We know they're doing a lot of important stuff, but why do they have to trim themselves now? And so those are things that we're interested in terms of development.
So there are still some questions, but I think we are already over time. So yeah. I guess we have to conclude here. Again, I want to thank you again for your enthusiastic talk and really the amount of data and unpublished data for sharing all of this with us. So thank you again for the probably more than 200 people that are following this. At the moment, they are currently about 200, but there are also other channels. So again, thank you so much for participating. Actually, it was again fantastic. We really reached. We were going from India to Peru to Brazil. Oh, wow. And back, and so really showing the power of going worldwide. So and sharing that your science with everyone. So thank you.
I really appreciate this, Gaia, and I really appreciate the questions. Again, some of this stuff is ongoing, so we welcome feedback and suggestions. I should mention, I didn't have this on my slide, but I should mention that we're also on Twitter, so you can find us at AO Lab. If you want to reach out to us and contact us. I appreciate it. Thank you so much.
Thank you again. Have a good day.
Okay.
Bye.